So we are here for the next of our panel sessions. I'm Madeleine Murtagh. I'm co-chair of the Regulatory and Ethics Workgroup. I'm Professor of Sociology and Bioethics at Newcastle University in the UK. So despite the accent, it's the UK version, not the Australian one. So one of the things that's become really clear over this meeting is that the zeitgeist, if you like, of this meeting is in fact diversity. We heard the brilliant talk yesterday from Julia Stojanovic, who was talking about the ways in which inequalities, lack of diversity are built into the data without us even realising that they're there, and we have to be really mindful of that. In the Genopri workshop that we had yesterday, Mwenza Blel reminded us that in discussing and thinking about privacy, we need to think about privacy for all, and we need to remember those histories in healthcare and research that don't necessarily take account of diversity, inequalities, etc. So in this session, we have three speakers who will explore the challenges of ensuring diversity in a beautifully GA4GH type transdisciplinary way. And first of all, we have, my list is in the wrong direction. We have Calvin Ho, who's a lawyer by background. He is assistant professor at the Centre of Biomedical Ethics in the Yong Lu Lin School of Medicine at the University of Singapore, National University of Singapore. He's got a lot of accolades. World Health Organization Collaborator Centre on Bioethics in Singapore, member of a number of ethics committees, Médecins Sans Frontières, Singapore Medical Association, on the board of the Legal Aid Bureau in Singapore, and his work examines governance, health policy, bioethics and law. So I give you Calvin Ho.
Thank you very much, Madeleine. As you may have heard from the introduction, I'm quite an indecisive person, hence my interest in the topic of AI. Perhaps that might help me. So I'm just going to... Yes, okay. So thank you again, Madeleine, for this opportunity to speak. So this is essentially my presentation in a nutshell. I've promised to keep to 15 minutes. So essentially, I thought AI being such a broad topic, I will focus in particular on its applications in a healthcare setting. So I'll tell you which one in particular, a particular application that's recently been approved by the US FDA. My sense is that looking at AI applications within this domain, there has been no new ethical, I would argue, legal issues. However, I think that many of these issues are similar or common with some of the technical challenges that we see. But I also think that a lot of these issues are likely to aggravate some of the existing challenges that we're confronted with. My main concern, quite similar to what Julia has mentioned to us yesterday, is a deepening inequity that we are beginning to see, and that is going to worsen over time if nothing is done to arrest this progression. There are at least two important responses to my mind. There are more, of course, but I thought for this short presentation, I'll speak on regulatory safeguards that need to be in place here. I thought that some things that have been shared with us on day one of this particular meeting by Tad Dove on regulatory stewardship is quite helpful to build on. And then yesterday, of course, we've heard about the issue of equity, particularly in relation to race and gender. So that is one other aspect that I think we'll need to think about. And then finally, I'll try to bring it all together in terms of thinking about how we could draw on some of these technologies that we're seeing on the horizon to construct a responsible and responsive learning healthcare system. I'll explain a bit about what this means, but what's critical for this particular enterprise, to my mind, is that it must be participatory and it must be transdisciplinary. So this also building on what we've heard yesterday from Madeleine and Mavis, as well as Anne and Richard. So the technology that I thought I'll focus on, the application, the AI application in particular, is IDXDR. Some of you may know this as a software that's been recently approved by US FDA. And this is essentially used to diagnose mild diabetic retinopathy for people who are already diabetic. But of course, what's important here is that you see that's where it can be entrusted with this task with very little or no physician input. So that's where we already see a sort of independent use of AI software within a particular healthcare setting. So some of you may have also seen this particular publication. So this has been the study that provided enough evidence for FDA to feel confident enough that it can be approved for use. But of course, there are a number of important limitations here. In terms of the training data that's being used, it's still rather limited. So as far as AI goes, as everyone in the room here knows, I believe that there is a high dependency on quality and the amount of training data. Data generation and curation continue to be challenging in a number of contexts, in healthcare in particular, I would think. There is also, if we are to scale up on this particular use of IDXDR, there is also the problem of explainability. So currently, of course, we don't have this particular problem with this software because the deep learning aspect has been locked. So if we are to activate or allow the deep learning process, and that's where you find value in this particular software, we are going to be meeting head-on this problem of explainability, and that, of course, is the black box problem. There are a number of continuing legal and ethical issues, and now, of course, contained within this limited setting, but if we are to put this particular device out there for general use, then there are a number of questions that we need to think quite carefully about, particularly in the sort of distributed agency that you'd find, because the responsibility then is not solely in terms of the clinician who has responsibility for this particular individual, but also somewhat shared with this particular software device. There are serious concerns with bias. I will speak a bit more about that, and possibly also with over-reliance in terms of how healthcare workers will respond to this particular device when scaled up in terms of application. And there is also, as yet, uncertain impact on healthcare workflow. We have seen a lot of very good social science research in relation to telemedicine. So we will have to replicate this sort of research in order to better understand the impact of such technologies in terms of the day-to-day healthcare practice. So as I've mentioned, no particular surprises in terms of the legal and ethical issues that we're faced with. As I've mentioned, it amplifies some of the existing problems, but I do think that the challenges surrounding AI, so when we think about trustworthiness, explainability, bias, vulnerability to attack, all of these are not merely social and political issues, but also technical ones. So very closely linked to the sort of things we've been talking about in the past few days. And I think hence very timely to consider what Madeleine has been speaking about in terms of transdisciplinarity. So some of you may have read this particular work. So Eric Topol, he has a rather more optimistic assessment of the impact of AI on healthcare provision. I'm somewhat more skeptical. Of course, to his mind, and I think quite rightly, AI is likely to and foreseeably replace some of the more mundane kind of work that we see within a healthcare setting. And of course, he feels that with this particular development, clinicians will then have more time to spend with patients and develop perhaps other softer skills, including like empathy. So my sense is that if we are just to leave the situation as it is without any sort of systemic arrangements being put in place, and to better prepare our healthcare providers in terms of these sort of technological developments, what we are more likely to see will be growing inequities that Julia has mentioned to us about. So, for example, if we are to look at this particular facial recognition technology, we already know that there is a problem with reliability and bias. Of course, in terms of this particular face-to-gene recognition app, what this particular study has found would be, of course, serious concern with racial discrimination, continuing discriminatory biases being built into the system. A number of you may have also looked at the report of the American Civil Liberties Union study on the Amazon's app recognition. Of course, what they've done here was to attempt to relate this particular app, apply them to the members of Congress, and to see and to compare it with, of course, individuals with a criminal record. Interestingly, of course, it has been shown that there's been a wrong pickup of more than 20 individuals indicated as having some sort of criminal record. So that goes towards the lack of reliability. And about a quarter of these individuals are from, of course, non-Caucasian racial background. So that's where we have some of the continuing biases that we see in applications such as this one. So my big concern is that with a lot of the competition that we see in the field and a number of problems that we have already heard over the past few days, there is more likely to be deepening inequities unless we take some serious measures to address these particular concerns. So this brings us back to perhaps the all too familiar tussle between equality on the one hand and efficiency on the other. So a lot of the AI investments have been directed at building or harnessing the sort of efficiency that we hope for. But perhaps a long time ago, we have already heard from this or seen in this particular publication that unless there are particular safeguards to find a suitable place for the application of this sort of technology and also to set clear boundaries around it, we are likely to be confronted with a lot of problems. Now this is not an easy task, as I've attempted to highlight here in a publication with some of my medical colleagues. We find a representation in these technologies of only certain values but not others. So what we would see is essentially a sort of value colonization where some of the other more important values are being crowded out, so to speak. So there is a need to build in the sort of value basis and to diversify the value platform to which these particular technologies are being constructed upon. And for this to happen, we do need regulatory intervention. We have seen this in the GDPR. Of course, Article 22 of the GDPR requires explainability. But of course, this in itself could create other problems because when we talk about explainability, then to what extent should it be explainable? What exactly should be explained? And how clear should it be, especially in terms of very sophisticated applications? So we are confronted with a number of these issues, but at least at a starting point, we do need a regulatory platform that can bring together a broader kind of value system. But of course, if we do broaden out the value system, it does create other issues, because when we think about equality, there are actually quite a few ways to understand it. So are we talking about equality? Are we talking about equity? Are we talking about social justice? Are they all the same thing? In ethics, not quite. So we haven't got a unity of value as it is. So if we think about just by design or try to build in justice, then we need to think about what exactly do we mean by justice? Are we talking about justice in a utilitarian kind of way? Are we talking about a justice that perhaps might promote certain types of liberties over others, assuming that we can speak of equality and liberty coherently and in a consistent manner? Are we going to emphasize communitarian priorities? How about egalitarianism? So quite a number of contentions. And if we think back about what Julia shared with us yesterday, within a legal setting, of course, there are other notions of justice that need to be accounted for, particularly those that are retributive and compensatory in nature. So it's quite a complicated picture. There have been quite a number of discussions around moral machines, which means essentially building in a moral sense within the sort of applications or devices that we build. But Isaac Asimov, a long time ago, with the famous fundamental laws of robotics, will show us that ultimately this will not be good enough. And one of the problems is with value alignment. As we have seen, we do have a lot of different values that are in place. The various apps can represent different values, and I do think this in itself is important. But it does mean that we cannot then rely on these devices to make moral decisions, because ultimately when they come into situations of moral dilemma, moral distress, or conflict, it will just enter into paralysis or become neurotic, Or perhaps in some of the sci-fi movies that we've watched, they might become totally neurotic and become a threat to human existence. So we're seeing that this is difficult, but we are still attempting to draw in. Oops, sorry, gone a bit too quick. So yes, this is my last slide. So well within time. So what we ultimately do want to try to advance, drawing on the sort of regulatory and governance mechanisms that we want to set up, and also being aware of the sort of justice concerns that are in place, and the measures that we need to put in to actively promote some notion of justice. We need to bring these together. It cannot just only be in the machine or in the device. It must complement systemic arrangements that are currently in place, and I think that even at the systemic level, a number of fundamental changes need to take place, and of course, this can only happen over time with appropriate evaluation, monitoring, and of course, adjustments in place. We do not as yet have on the ethical or legal front any sort of mechanism that supports this sort of work. I think moving forward, we do need to think very carefully about this particular space. And here we need to work collaboratively with the technical experts. So because these issues and these challenges are shared, I do think that there's a lot of room for transdisciplinary work. And again, I'm very grateful for this opportunity to start the conversation at least. So with this, thank you very much for your attention, and welcome any questions after this presentation. Thank you.
Thank you, Calvin. On the topic of questions, I've been asked to ask you all if you might think about putting your name onto the Slido when you ask when you ask questions. You don't have to, but it'd be quite nice to know who's asking. So the next speaker is Alicia Martin. She's a population and statistical geneticist examining the role of human history on global genetics and global genomic and phenotypic diversity. She has posts all over the place, Massachusetts General Hospital, Harvard Medical Center, Broad, and her work looks at the generalizability of genomic knowledge. And excitingly, she's developing statistical methods and resources for studies of diverse and underrepresented populations. Thanks, Alicia.
All right. So I hope you can all hear me. My voice just returned a couple of days ago because I've been a little under the weather, so please raise your hand if you can't hear me at all. So it's really a delight to be here. I love this forum, and I love all of the activities that you guys have been putting together. My background is a little different from Calvin's. My background is more in the population genetics, complex trait genetics, and bioinformatics space. So I'm really excited to talk about the clinical use of current polygenic risk scores and the need for diversity when thinking about how genetic technologies are going to translate across a very diverse spectrum of individuals. So first, to get started, sorry, as many of us in the room I think are very familiar, genetic variation reflects human history. If we look at the DNA of anyone in this room, we can see your ancestors in your DNA. Humans originated in Africa, on the order of hundreds of thousands of years ago, and then migrated out of Africa into the Middle East. From there, migrated into Europe, and then into East Asia, into the Americas, and spread all over the world. With each of these migration events came a subset of the genetic diversity that originated in this starting founding population. So as we look at populations that have moved outside of Africa, they have a subset of the genetic diversity in Africa, and as people migrated throughout the world, they took a subset of that genetic diversity with them through these serial founder effects. So it's been really exciting to watch as GWAS have grown increasingly powerful. We've learned a ton about human history through genetic data, also through anthropology, archaeology, neighboring disciplines. This interdisciplinary nature has taught us a lot about human history. But as GWAS have grown increasingly powerful, we've also translated this knowledge into our understanding of biomedical traits. So I think this forum is all very familiar with the fact that genetics has been growing explosively. We can see that just in the past decade or so, we've had GWAS being run on the scale of millions of individuals now, where it was only hundreds or maybe thousands of individuals a decade ago. But as we're also very familiar with, genetics has a huge diversity problem. So if we look at this slide, you can see that the representation of participants in GWAS are far out of step with the global population. So in the global population, for example, about 16% of the world has European ancestry, whereas right now, if we look at the representation in GWAS, about 80% of participants have European ancestry. So clearly we've got a lot of room to grow. And in looking at these trends and examining these trends over time, over the past decade, we've seen some problematic trends. So we've seen that, for example, the fraction of participants in these large-scale genetic studies started to diversify until around 2014, and since around then, progress looks like it's roughly stalled, or perhaps those European ancestry studies have accelerated in sample size, whereas non-European sample sizes haven't gotten substantially larger over time. So this is a big problem in the field that we're trying to grapple with. So the central question that a lot of my work tries to address is how these ancestry study biases in genetics actually impact the generalizability of this knowledge. I think there's a few important considerations that we need to keep in mind here. One is that we expect the fundamental causes of human disease, the genetic causes, to be really consistent across populations. We've seen this in all studies that have done fine mapping and looked at the effects across populations. By and large, there's very little heterogeneity in effects across populations. So the very fine-scale biomedical pathways that are impacting disease in different populations seem to be shared, and that should be, I think, common sense. But we know that human history is in our DNA, so allele frequencies are different, linkage disequilibrium is different, so the correlation structure across the genome, environments are different, and many other factors like natural selection, those exposures and how the genome responds to environment may differ across different populations due to their different population histories. So that can create challenges here. So a couple of years ago, we wrote this paper where we were looking at the effect of human history on genetic risk prediction across diverse populations. And one of the key takeaways that we learned from this study is that genetic risk prediction, the accuracy of this decays with increasing genetic divergence between our discovery data sets and our target data sets. In other words, if we're to do these large-scale studies in European populations over and over again, the more genetically distinct you are from a European ancestry population, the less accurately we're going to be able to predict any of your disease risks or your traits. Anything that we're trying to predict using genetic data will perform worse the more genetically distant you are. We also learned that polygenic scores can be different across populations arbitrarily, but these biases are not necessarily meaningful. So if we look at this slide, I've computed polygenic scores for height in the, using data from the GIANT Consortium. The GIANT Consortium is aptly named. It's a very large consortium that's looking at biomedical and anthropometric, primarily anthropometric traits like height, BMI, waist-hip ratio, those types of traits. And they've done this primarily in European ancestry populations. If we compute polygenic scores for height across these globally diverse populations from the 1,000 Genomes Project, what you can see is that European ancestry populations are predicted to be the tallest, followed by admixed American populations, including Hispanic and Latino populations, followed by South Asians, and then the shortest populations are predicted to be East Asians, but not much taller are predicted to be African ancestry populations from West Africa. And these differences in these distributions are huge. So we're looking at like 5 standard deviation differences between these populations. So if you were to put some rough numbers on that, and you expect the average European ancestry man to be something around on the order of 5 feet 9 inches tall. Sorry for those on the metric system, I don't know how to convert this. And then you ask what a 5 standard deviation difference would be for African ancestry men. You might predict that the African ancestry man would be on average something on the order of 4.5 feet tall, which is shorter than me. So clearly this is far out of step with reality. So we know that we're having modeling issues with these polygenic score discrepancies that are simply arising due to stratification in our GWAS and the lack of consideration of different ancestry groups in our discovery cohorts. We also know that neutral human evolution alone is sufficient to explain these. You don't need to invoke any natural selection arguments to come up with these kinds of trends. It's simple drift. Last year, or earlier this year, some of our colleagues recapitulated some of these results. So here's a, I think, nicer map than our study put up, showing what predicted height looked like in globally diverse populations around the world. Again, you can see that European ancestry populations were predicted to be the tallest, and the shortest were predicted to be East Asian and African ancestry populations. So this is what they see from the GIANT Consortium. Looking at data from the UK Biobank, which was collected in a more harmonized way, a key principle of this particular group, you can see that there are no predicted differences across these populations when put on the same scale. So clearly we've got actual differences in populations. Some of these may be genetic, some of them may be environmental, but our discovery cohorts are not letting us identify differences within and between populations, and I would suggest that it's premature to try to be doing comparisons across populations using something like polygenic scores. We just don't have the methods to do that right now. Some of our colleagues in Finland have really tried to drive this point forward and looked at a finer scale level of polygenic prediction accuracy. So Finland is a particularly interesting population for a couple of reasons. One is that it's a particularly bottlenecked population. It's relatively homogeneous and isolated, but there have actually been serial bottlenecks that have happened within Finland. So the southwestern part of Finland is more cosmopolitan, and there's been a serial bottleneck moving into the northeastern part of Finland. So there are multiple bottlenecks that have happened there. It's also clear that there are height differences in Finland. So the southwestern part of Finland is on average about 1.6 centimeters taller. And when we look at polygenic scores for height, just trying to predict this simple trait that's very easy to measure, using 3 different types of data, using the giant summary statistics, using the UK Biobank summary statistics, and then also using a Finnish set of summary statistics, what you can see is that the predicted differences are very substantial between these different cohorts. So we're predicting a 3.5 centimeter difference using the giant data, only explaining 14% of the variance. So we're already vastly over predicting the difference, especially if you scale this up and we're able to explain all of the variance. In UK Biobank, we're doing about on track with what we would expect. So we're predicting a 0.6 centimeter difference, explaining almost a quarter of the variation in height. And then with the Finnish data, which is actually much smaller, there's only about 25,000 individuals. These are an independent sample. We're also over-predicting the difference here, despite processing the data in a relatively homogeneous way, like with UK Biobank data. So this is I bring this up mostly to raise the fact that there are a lot of considerations that we don't fully understand. We don't necessarily know why we're over-predicting differences when we have an ancestry-matched cohort here in Finland, even when it's substantially smaller than the UK Biobank data, which is processed in a harmonized way. There's still a ton of open questions to try to investigate here, and it's really premature, I think, to try to be implementing a polygenic score in a clinical setting when we don't necessarily have a full handle on that, even within a relatively homogeneous country like Finland. And that would be, exacerbated here in the US, where we have a ton of ancestral diversity across different populations. So earlier this year, we also looked at how prediction accuracy differs across globally diverse populations that are present in the UK Biobank data. UK Biobank is this wonderful resource of 500,000 individuals that's primarily European in ancestry. About 95% roughly of individuals are of European descent. But 5% of 500,000 is still a pretty large number on the scale of things, so we actually still have a pretty non-trivial number to look at in this UK Biobank that have not really been analyzed to the same extent as European ancestry populations. So to try to investigate questions of polygenic risk prediction accuracy across globally diverse populations using one of the largest data sets out there, the UK Biobank, we looked at prediction accuracy for 17 different quantitative traits and asked how well we did in European ancestry populations relative to other globally diverse populations. And we identified pretty staggering disparities in the prediction accuracy. So if we normalize to the European accuracy, so we set that at 1 and ask across these 17 different traits how we're doing across these populations, you can see that the more genetically divergent you get, the worse you do, and that these differences are very non-trivial. So in East Asians, for example, we're doing about half as well as in European ancestry populations. And we're only explaining about 20 to 25% of the variance in African ancestry populations as we are compared to European ancestry populations. This is predictable. We should have understood this going into this. So the genetic basis of this is pretty, I think, clear. We're building polygenic scores from correlated variants, not necessarily causal variants. We don't know what those causal variants are, so we can't build polygenic scores from that. And there are complicated differences on the genetic end. So there are differences in allele frequency and linkage disequilibrium that are going to drive some of this. The rest of it is harder to pin down. There might be differences in natural selection. There are other complicated gene-environment interactions happening here. There are a whole slew of reasons that this might be more complicated than just these simple genetic factors. So I'm going to skip this for the sake of time, but basically we should have known that this was going to happen. So we've gone through and compared the UK Biobank with the Biobank Japan, these two very large-scale data sets. We wanted to understand whether there was promise coming from these diversifying efforts. So to try to answer this question, we ran equal-sized GWAS in UK Biobank and in Biobank Japan for overlapping traits, and then computed within and cross-population prediction accuracy, and asked, do we see symmetric and comparable polygenic risk score accuracy for these different traits? I should mention, I teamed up with a really talented grad student to do this, Masa Kenai. He's wonderful. So these are the crux of our results. So the different colors in each of the plots represent where our summary statistics are coming from. So in blue, we have our summary statistics are coming from the East Asian discovery cohort, and in red, we have our prediction is coming from the European UK Biobank samples. On the y-axis in these two different panels, these are our different target cohorts. So on the left, we're predicting in European samples. On the right, we're predicting in Japanese samples. And you can see that for all of these traits, we're doing better with ancestry-matched results. So on the left, we're doing better predicting in the European populations using European discovery cohort. On the right, we're doing better in the Japanese cohort using the Japanese summary statistics. But you can also notice that the y-axes are quite different here. We're not actually doing similarly, despite having set this up in as controlled a manner as possible. In other words, we're predicting the European traits about twice as well as we are in the Japanese traits on average. And the reason for that comes down to a cohort dependency. So UK Biobank was set up to measure these quantitative traits. Biobank Japan was set up to measure disease traits. And so we actually do better at predicting diseases in the Japanese cohort and quantitative traits in the UK Biobank cohort for those traits we analyzed. I'm going to skip this for now, but I just want to mention this is true for a bunch of quantitative traits. It's also true for things like schizophrenia. When we have ancestry-matched results, we're doing better in all cases, even when those sample sizes are a fraction the size of the largest European ancestry cohort. So I think an important point to remember is that we don't need our sample sizes to be equal across all different groups. As we start building these, we can aggregate them and have improved prediction accuracy for all populations and underrepresented populations, building on some of the previous progress. So where do we go from here? One area I think we really need to be focusing on, and it's great to see our H3 Africa colleagues in the room, is to really focus on those populations that are the least represented in genetic studies. So we've been trying to address this by focusing especially in Africa. So there's a project called the NeuroGap Psychosis Study, the Neuropsychiatric Genetics in African Population Study. This is on track to study about 35,000 African populations across Ethiopia, Uganda, Kenya, and South Africa. Of course, there are H3 Africa efforts to build large-scale biobanks as well. Alongside, and hopefully in partnership with many of these colleagues, we're building genomic resources for African ancestry populations, including, for example, imputation panels. And we're trying to make sequencing versus genotyping recommendations for these new studies. We're also building capacity through in-depth training programs. One program I'll mention just very briefly is the GINGER program, which it's a 2.5-year program consisting of on-site workshops, virtual classrooms that happen weekly, and on-site trainings that are tailored to each of the research institutions. I think it's very, very important that when we try to expand the diversity in our genetic studies, we're doing so in an ethical manner that brings in a sustainable workforce that can actually contribute longer term without the need for investigators in high-income countries. So with that, I just want to say I think our next steps are that polygenic scores may be very useful in social sciences as well as in biomedical studies, and they may improve things like clinical models, but right now I think they're really likely to increase health disparities due to Eurocentric GWAS biases. To ameliorate these, we need more diverse GWAS studies and new methods to address these major issues. And I would just also push the idea that we need to be really careful about how we're communicating our science, especially when our research touches on culturally sensitive topics. We need to anticipate the implications of our research and communicate these widely. I'll take questions after our next speaker. Thank you.
Thank you, Alicia, for that fascinating talk. So our next speaker is Mavis Machirori, and she is a sociologist and anthropologist with a clinical background. She's a postdoctoral researcher at Newcastle University with me, She sits on the diversity panel of Genomics England's Ethics and Social Science domain, and her work examines the views of people in low middle income countries and minorities in the global north, and how those diverse populations, how those views feed into the governance of genomics. Thank you, Mavis.
Thank you, Madeleine. Hopefully everyone can hear me. So this conference has been really interesting in terms of speaking about diversity in research and in our participants, but also this has been spoken about in Genopre in the last 2 days. It's not something that is particularly new because now, if this works, literature supports this. We've already heard this from the previous speakers that there isn't that much diversity. If you look at GWAS studies and the individuals within those studies, most of the people are of European descent. But it's not just about patients and publics, it's also about the right workforce for these kinds of studies, especially for genomics and genetics. Discussions of implications of genetic tests, for instance, whether clinical or research, need trained personnel, such as genetic counselors, and yet when we look at the map on the left, most of the genetic counselors are actually across America, Europe, and Asia. So this asks the question, who's talking to people about genetics and genomics in African populations, for instance? Additionally, when we look at Toland's map on the right, in 2013, they did a survey of labs that were undertaking BRCA, which is the breast cancer gene testing across the world. And of the ones that responded, most of those labs were also particularly concentrated in in the global north. And again, their map shows this snapshot of lab testing. And although not every lab responded, it does beg the question of, if most of the knowledge is coming from labs in the north, what does this say about the knowledge that's produced and the claims that are made and produced in those areas? So when our research doesn't address some of these issues in resource distribution, for instance, I believe the importance of our work will suffer in the long run. And while we strive to increase representation in genomics, as Calvin Ho and Mwenza Blau told us in Genopre, we have to reflect on how our activities, whether this is knowledge, production, or the claims that we make, can actually cause harm to patients and publics, especially underrepresented groups. Even not researching concerns of certain populations can also be harm because this will lead to greater health inequalities. So when we think about our research participants and increasing diversity, which is a good thing, what else should we be thinking about? What do research participants say about our past and our current broad and medical general genetic practices? I will highlight some of these concerns, showing that taking scientific practices without the social context only hides the sometimes everyday concerns by seldom heard voices within research. To do this, I will use some quotes from Black women and Asian women I spoke to during my studies around familial cancers. I'll also use some of the narratives I've seen and heard on social media and in literature. So one of the questions we ask is diversity a social problem or a medical problem? And of course, as a social scientist, my answer is it's complex. In trying to answer, for example, why Black men and Black women have higher mortality rates for prostate and breast cancers, headings such as these tell us that if we actually if we ask the right kinds of questions, then we see how good genomics is. But what happens is that until these kinds of specific types of knowledge are actually available, people's concerns are usually not addressed. And so it means that in the end, the experiential knowledge is disregarded and the concerns are disregarded. And because there's no scientific medical evidence, then there's no clinical pathways for people. This is difficult for minority patients because if their experiences are not answered by biomedical knowledge, there'll be no medical guidelines to help with diagnostics and treatment, meaning that people will continue to suffer disproportionately within health. And we know that because of complexities within social within our social communities, an equal burden of disease is actually also going to translate to an equal burden within economic costs. And as somebody said to me, it shouldn't take 2 or 3 members of the family to die from that illness before someone says, well, let's look at it. So the guidelines that we use to care for and treat patients influence patients' interactions with health and research. But if we're saying, sorry, we have no answers for you, it's not surprising that the frames people use is actually what they will use for understanding their ideas of disease. For instance, when asked about what she thought of an inconclusive genetic test for breast cancer, given her personal and family history of disease, my participant said, the test didn't mean anything, apart from the fact that I feel, okay, we're in the Western world, and they've got more information about white people than they have of us black people. So no information or lack of information of underrepresented people can lead to questions about how useful medical knowledge is. People can then withdraw from participating in research because of them not seeing actually the utility of that information for people like them. But we also have to remember that sometimes it's not just about medical knowledge. It's also that, the circumstances in which people come from can also frame the way that they see the disease. So another woman said, if you're an African, you don't have money to be treated, obviously you have your faith and nothing else. So if there's no testing available, you won't care about it really. And if you remember the first maps, where a lot of the labs might be concentrated within the north, if those tests are not actually being performed within local areas, then people don't actually have the opportunity to interact with that kind of knowledge or that disease. So not participating might be weighing up questions of knowledge versus ways to cope, which are themselves framed by resource availability or lack of. So as I said earlier, the picture is complex. Yesterday, Julia, in her keynote, showed us how biases can increase inequalities and that things are actually more connected than we sometimes realize. So controversies such as the Tuskegee trial, for instance, and perceptions of research misconduct or seeming lack of transparency, and here think of the biopolitical responses to the Ebola and the Ebola vaccine. These all create a whole extra lens with which Black people, but also other minorities and indigenous populations in the world, view medical practice. I saw something on Twitter when people were talking about why Black men, for instance, in the States don't go to see their GPs. And this woman said, it's because we walk in the ghosts and shadows of those that have gone before us. And what this means is that if there's no transparency in the way that work is done, people would judge you and your intentions by the histories that form part of their own identities. And so in that case, they will not participate in research. And I have a quote again from one of the women that I spoke to. Although this was a discussion about cancer, actually she still brought up things about non-cancer related concerns, in which she says, imagine, Ebola has been there for ages, for more than 30 years. Nothing's been done about it. But because 1 or 2 people from Europe have been touched by it, they managed to find something to kill the virus. So what can we say what can we think about when we see things like this? So although in the medical world, the way research is done isn't always perceived in that way. We have to think about, as the previous speaker said, the claims that we made and how we make those claims and the timelines, because in emergencies, things take on different meanings. So we have to ask, is our work perpetuating previous wrongs or building trust? Because if it is perpetuating previous harms or recreating those kinds of concerns, participation by minorities will remain low. And so is research at the moment different from what it was in the 40s, 50s, 60s, or 70s? And what I've hopefully shown in this short space is that actually participation, representation, and inequalities all tie in together. Because if we don't do something about widening participation and diversity, there'll be judgments on the research that we do, and there'll be no or misrepresentation in research, and this cycle will continue. So it's not all grim. The solutions are actually present, and I think we're all in the same, in this room, are able to contribute to that. I believe that if we work in multidisciplinary ways and include ideas about sociopolitical ideas about identity and related classifications that we use in research, then we can start to tackle some of these concerns about genomics diversity. For example, what does genomics research do to cultural groups and their geographical boundaries? What impacts go beyond disease and that are political in the ways that we define genomics? And how are worldviews and concerns recognized in our consenting processes, for example, or the questions that we ask in the data? We talked about public engagement yesterday, so we have to be creative about the ways in which we speak to people about genomics and go beyond just educating about genomics knowledge itself. We also have to think it's not only just going to be on patients and publics, but colleagues in research as well. Underrepresentation in STEM subject areas means that it impacts the types of questions that we can ask, the types of questions that we can ask, and the diversity that we then have within that research, and ultimately how useful the knowledge that we generate is. So if we're going to talk about diversity, we have to give space to change our practice as well. And finally, to summarize, as we move to understand diversity, we must not make people the cause of their diseases when we've neglected them in other areas and in health for so long. Who are we using as the reference for what we classify as normal? What social contexts are we missing in asking our research questions? We have to be creative in our approaches and remember that history is not always written. So lack of participation is not always going to mean a lack of interest. And I think we have a duty to do better. Because is it really reasonable that people should have to wait 30 years to get meaningful information from our current biomedical technologies? Thank you.
All three of you for a fabulous set of presentations. And thank you for sitting on the side where there are three mics. It's going to make it a lot easier. So we have a short time. We have 7 minutes for questions. I think the lovely blue things that will cast your voice around, so if you feel like actually standing up and asking a question, just put your hand up or stand up and varying people will come to you. So going to the first question from Ewan, should we move beyond these broad ancestry group definitions in genetics and recognise the complexity of human population histories with admixture? I think each of you will have a response to this. So we'll start at the end. Mavis.
Okay. I think I don't know that I can have a straight answer, but we have to recognize that in terms of, for example, on the African continent, boundaries within countries were very just kind of, we're going to rip up a map and give you this section. And we have to think about how those kinds of boundaries map or don't map onto how people define themselves and their identities. And I think we need to move beyond just group definitions, because actually in terms of the things that matter in genomics, which are also the values, we have to try and map the social to the science. And I don't know yet that there's a right way of doing it, but we need to listen to what people say their identities are, because actually sometimes the identities go beyond what we think is classification as Black African or Black Caribbean. So it depends on where people map themselves onto, and maybe that's a starting point to say, well, what actually matters to you, and how did you get to where you are, and where can science actually then answer those kinds of questions? I think.
Alicia?
Sure. Yeah, so I totally agree with everything you said, and then would also add, I think when we're looking at genetic data, often we're looking at ancestry. Ancestry doesn't mean anything for one individual. It only means something in comparison to other groups. So it's really hard to say, like, what my ancestry would be or what your ancestry would be. We're looking at like several different cultural and scientific terms here. So we're looking at the interplay between genetics and social identity. So we're looking at the crossplay between ancestry, ethnicity, race. All of these have slightly different meanings. They might be correlated in some way, but they're not necessarily going to be the same thing. So I think I totally agree with you that we need to be considerate of the fact that there are very, very many ethno-linguistic groups. There are very many ways that people are going to self-identify, and this complexity of human history has a lot to do with how people are going to self-identify, but it doesn't always capture that. So I completely agree that we're going to need to consider the complexities of human history and the social identity of people in genetic studies.
Yeah, thank you. I think from a value standpoint, it may be helpful to think quite critically about why we're asking those sort of questions. And in terms of the information that's being generated, what are some of the normative implications? Just to pick up a point that Mavis mentioned, are we actually perpetuating some of the inequities that we already find in the system? Are we sustaining particular issues in a way that does not help address some of the deepening inequities that we see today? So what exactly, I think, ultimately is the role of scientific work, and where, again, does it interface with the broader social aspects in relation to the work that's being done?
Thank you. The next question is a very important one. What if minority populations refuse to share because their researchers may not benefit from that research? People are afraid of academic colonisation. Somebody like to respond?
That's deeply political, isn't it? I think it's really difficult. At the moment, a lot of knowledge has gone from south to north, as in samples and information. And there's a very real reason why people might be worried about sharing their data. And I think similar to what both Calvin and Alicia are saying is when we're doing this research, if we're building capacity, it's not going to be, let's build capacity, let's collaborate, but then we take the data away. It's actually, well, let's put labs on the different continents. Let's create equality in terms of how samples are then utilized. And until I think we get representation of people producing the information, a lot of people will probably be quite reluctant to share their data. And we have to work, it will take years, I think, to build this kind of trust, and to remember that actually things in the media sometimes will break that trust really quickly, which is why I think it's more complex than just let's share the data. Let's think about the historical reasons why people might not want to share their data.
Yeah, I just want to support what Mavis has mentioned. I do think it's very important to build community trust and to have a collaborative relationship with communities that are affected by the research. I think we've seen this recently in relation to, say, for instance, the Ebola outbreak, how important it is to have community engagement, to sustain trust. And it's interesting to see that perhaps from the angle of international law, we have the Convention on Biological Diversity and the Nagoya Protocol that's intended to facilitate data sharing and sharing of materials. But then what we find that's being used is these resistance and these walls that are being constructed through some of the, I think, more intricate legal practices. Why that is happening is, to my mind, one that is a lot deeper in relation to benefit sharing. Of course, we've talked about. There are these communities that are, I think, generally concerned about what ultimately comes back to them. Are they just essentially being used as guinea pigs? And as researchers, we do need to think ultimately what are some of the longer-term implications in terms of trust and the science that we do as a moral enterprise, not merely as a technical endeavor.
Thank you. There's a nice positive question from Ashley, wherever you are. What are some of the positive examples of building trust within, and presumably between populations, underrepresented populations? Would anyone like to take that?
I think, there are countless, in just thinking about some of the collaborative work we've had with researchers in the psychiatric space, coupling European ancestry data with these large partnerships spanning Eastern and Southern Africa. We've already learned about some of the more important environmental contributions to disease in advance of doing any genetic studies for some of these underrepresented populations traditionally that are, I think, going to contribute to global knowledge. just in the, setting up of this project, without even necessarily getting all the genetic analysis done, we're already learning about some of those sort of psychosocial factors that can be culturally very important and meaningful. I would also mention that some of the drug discoveries that are being made as a consequence of genetics are coming because we have underrepresented populations included. Thinking about things like PCSK9 loss of function variants that lead to these new drugs that can lower cholesterol effectively in families that might not necessarily respond to your classic statins. Some of those discoveries came because African American populations were included in these studies, and they wouldn't have been identified as effectively or nearly as quickly, and we wouldn't already have a drug on the market that can treat some of those diseases more effectively. So diversity is critically important for everybody. We find these new drugs, we find new biomedical pathways. All of these things are really important and emerge from diversity. Additionally, we can pinpoint causal variants that are actually impacting disease most effectively by bringing together more diverse ancestry groups. All of these things are scientific benefits that don't happen in a relatively homogeneous bubble.
Thank you, and I think that's a great note to finish on. Please join me in thanking our speakers.